Okay, in this session I'm going to cover two different recent reproductive advances. I'm going to cover the three-parent fetus, which is a way of families suffering from mitochondrial diseases from having children which are biologically related to them but free from disease, and we'll see how that the procedures that we talk about today are essential for that to happen. And then we're going to touch on cell-free fetal DNA detection in maternal blood. And this is an alternative to doing an amniocentesis or chorionic villus biopsy, completely non-invasive, and allows us to test the effectively the genotype of the fetus from fetal DNA that is floating around in the mother's blood. So in the section on the three-parent fetus, we will be covering why it might be needed, so the very specific clinical need where this is performed, how it's done, and we'll also be looking at why other tests such as pre-implantation genetic diagnosis and prenatal diagnosis are completely unsuitable for this type of disease. They just don't work, but we will touch on that as we go through. And we're going to look at some of the potential issues with this, about whether we are manipulating the human genome doing this procedure or not, and look at some of the ethical issues associated with it. So firstly, we're going to cover why the three-parent fetus-based IVF is needed. And this is because most of the inherited genetic disorders are carried on the autosome, so chromosomes 1 to 22, and then a relative minority are sex-linked on the X and the Y chromosome. There are quite a lot of X-linked disorders, very, very few Y-linked disorders, but a further minority are passed on via defects in the mitochondrial genome, and these are what we call the mitochondrial diseases. So these are completely different to the classic inherited mutations. The mitochondria itself contains about 15 kilobases of DNA coding for a handful of protein coded genes and a full set of mitochondrial ribosomal RNA and transfer RNA genes, and they produce the ribosomal RNAs and the tRNAs required for translation of that handful of genes which are coded for by the mitochondrial genome. Most of the proteins that you find within a mitochondria come from the nuclear genes. So the vast majority of the metabolic enzymes are coded by genes on autosomes. mRNA is produced in the nucleus, that is exported to the cytosol, translated by ribosomes in the cytosol, and then those proteins make their own way to the mitochondria. So that's how the vast majority of proteins get to the mitochondria. But then this handful, half a dozen or so, protein coding genes are encoded by the mitochondrial genome, and they are transcribed from the mitochondrial genome within the mitochondria, and then translated using what we call mitoribosomes, which are ribosomes which exist within the mitochondria. So a typical ovum contains about 100,000 copies of mitochondrial DNA, whereas a sperm contains far less. So the sperm does not contribute really to the sperm mitochondrial DNA doesn't contribute to the fetus, but even with this small amount there, this should eventually add up to being, a fetus should contain paternally derived mitochondrial DNA. What I'm not going to particularly go into, but it does happen, is when if sperm mitochondria make it into the ovum at fertilization, they are selectively and deliberately destroyed, so that only the ovum mitochondria survive, and this is a deliberate process, and it happens across all species, that the sperm-derived mitochondria are deliberately destroyed by a very clever mechanism called the ubiquitin proteasome pathway that you'll learn about next year. So that is a deliberate way of getting rid of the sperm mitochondria, and presumably that is because the sperm are derived from rapidly proliferating cells, got a much higher mutation burden, whereas the ovum is very low proliferative, and the mitochondria, shall we say, much more genetically intact than the sperm mitochondria. So the end point of this is that the mitochondrial genotype of the offspring is determined by the mitochondrial genotype of the mother. And what we can see down here is two different family inheritances of mitochondria. Here's a male suffering from a mitochondrial disorder. When you've got a disease, we tend to fill in the square. Males are square, circles are female. So if it's filled in, this is a disease-affected individual. So in this situation, we've got a non-affected female. All of the offspring are normal because this woman is normal for mitochondrial diseases. Whereas in this situation, the male is normal, the female is affected. Every single one of her offspring is affected with the mitochondrial disease, potentially. And we'll come on to why it's potentially fairly shortly. So here we've got a couple of the well-characterised mitochondrial diseases: mitochondrial encephalomyopathy, lactic acidosis and stroke-like episodes. MELAS will do. So this is fairly late onset, and you get progressive stroke-like episodes later on in life leading to dementia. And then we've got Leber's optic, hereditary optic neuropathy. Now, with these two diseases, they tend to be present in the population of about 1 in 10,000. Certainly with Leber's, about 1 in 10,000 people have got the mutation, but only 1 in 40,000 seem to actually get the disease. And in clinical genetics we call this variable penetrance. And penetrance relates to whether the mutation, the presence of the mutation results in disease. And for some diseases, penetrance is low, and we covered this in the previous session on pre-implantation diagnosis. I think I told you that BRCA2 mutations, hereditary breast cancer, had about 50% penetrance. That means if you've got the mutation, you've got a 50% chance of developing breast cancer. In here with Leber's optic neuropathy, it's about 25% chance. So even though you've got the genotype for it, only about 25% of people actually develop the disease. And we don't entirely know why. We think that some people who are affected or have the disease state, only some of the mitochondria are mutant, some are normal, and it may well be just a proportion of mitochondria that are inherited with a mutation versus the normal genotype, and if it's predominantly mutant, maybe that causes disease. There's other things that are suggested to affect this, and it's possibly how this defective protein that causes Leber's interacts with other proteins, and if you've got genetic changes on other proteins that interact with this, then maybe that could make one person have a more severe type of disease, someone else have no disease at all. So it's complicated, but you just have to accept about 25% of people are affected. Now, in the previous sessions we looked at prenatal genetic diagnosis and pre-implantation genetic diagnosis. So the problem here is that all eggs carry the defect. So if we were to do pre-implantation genetic diagnosis from this female, every single egg would be affected, so therefore you can't do pre-implantation genetic diagnosis. Everything's going to be positive. Equally, you can't do prenatal diagnosis because every fetus is going to be positive. So because these two techniques don't allow you to select for a disease-free individual, then these can't be done, which is why we need something different. So the aim here is to allow a carrier or affected individual with one of these mitochondrial diseases to have offspring that are both genetically related to both parents and free from mitochondrial disease. So that's the aim of what we're going to be talking about. So we're going to have to somehow replace the mitochondria in an egg, or more correctly, swap the nuclei from an affected egg into a healthy egg that's had its nuclei, had the nucleus removed. So that's basically how the technique works. So we've got two different methods that allow this three-parent fetus to be produced, and the first one is what we call pronuclear transfer. So in step one, we've got two embryos. We've got the parent's fertilized egg, and we've got a donor fertilized egg. Now this donor fertilized egg has got healthy mitochondria, the parent's has got unhealthy mitochondria. So after fertilization, at step two, we take out the two pronuclei from this, we take out the two pronuclei from this donor embryo, remove that's removed and destroyed, and we implant these two pronuclei into this fertilized egg that has had its two pronuclei removed, and therefore when we implant these into here, we get an intact fertilized egg with intact mitochondria. So the key point here to consider when we think of the ethics of this is this method works very well, firstly, but it requires two fertilized eggs, one to act as the recipient for both the male and female genetic material, but it means you have a potentially unnecessary destruction of a fertilized egg, which ethically may be an issue for some people. So that's the main drawback of this particular technology. Now there is a separate method that we're going to cover next which gets around this possibility. So in this second method, which is what we call egg repair or maternal spindle transfer, we have the mother's unfertilized egg, we have a donor egg, and in this situation we take the nucleus out of the donor egg, and that's just removed and destroyed. We remove the mother's nucleus and we implant it into this donor egg. So we end up with an egg with the genetic material and the mitotic spindles and all of that machinery all together removed from this egg, put into this egg, and then what we can do is fertilize this egg with sperm from the father. So in both cases, this case and this case, we are using sperm from who we want to be the biological father, and we've got the nuclear material from the biological mother. The only thing that is donated is the mitochondria from a third party. So the key point here when it comes to the ethics of this is this may be more preferable to some people to embryo repair, as it doesn't create and destroy an embryo, only an unfertilized egg. So for some people from various religious backgrounds would see the creation of a fertilized egg as being the creation of life, and then destroying that would be the destruction of life, whereas this one here doesn't destroy a fertilized egg, so therefore ethically it is more accepted by some groups than the first method. So in the previous session we'd looked at other forms of genetic testing on embryos and fertilized eggs, and what we've learned about what can be done to an embryo or fertilized egg is that we know that selecting against genetic traits is okay. We know that sex selection in some disease situations is okay, and we know that selection on the basis of tissue type, which we learnt about last week, is also okay. What's not allowed is anything that affects the germline. So we are not allowed to correct genetic mutations by things like gene therapy and CRISPR technology. So that's currently not allowed. Now this puts this three-parent fetus in a bit of a grey area, because mitochondrial replacement does technically affect the germline on the basis that all subsequent generations will have a different set of DNA than what could have possibly been created in the first place, if you see what I mean. You are changing the DNA that embryo will have, in that you're replacing it with somebody else's DNA, albeit only a very small amount of it. Now, at this point here, what's not allowed is anything that affects the germline, as being redefined really as anything that induces a mutation in the germline, whether it's inducing a mutation or correcting a mutation in the germline or mitochondrial DNA, that's not allowed, but replacing the mitochondrial DNA is allowed. So this, although it technically is changing the DNA for subsequent generations, it is classed as not affecting the germline in the strictest sense, where it's not allowed. Okay, so this was approved in the UK in 2015. If you follow this link, you'll see the Human Fertility and Embryology Association site on mitochondrial donation and how this can be done and implications for parents if you want to read up on it. So both pronuclear transfer and maternal spindle transfer are both approved in the UK, and this is linked to the first case where a baby was born avoiding Lee syndrome using this 3-parent fetus or mitochondrial donation technology. So there are a few ethical issues that you might want to consider and have a little think about when we're looking at this sort of technology. Firstly, you think, well, who would you choose to have as the mitochondrial donor? We know that people are often concerned about designer babies. There is a slight possibility that someone who is in this situation would say, well, okay, well, I want to get rid of my frankly quite rubbish mitochondria and replace them with some really good mitochondria. Who would you choose to be the mitochondrial donor? So that allows some, very remote chance of some designer-ishness to the babies, but I don't think that's really become a great issue so far. The other issue is that if we're passing on a combination of DNA, mitochondrial and autosomal DNA that is going to be passed on to subsequent generations, then some bioethicists have proposed that maybe this technology should only be done and create male births, on the basis that male births would not have the capacity to pass on their mitochondrial DNA and autosomal DNA combinations to their offspring. If you allow females to be born using this technology, those females can be passing on a combination of autosomes and mitochondrial DNA, and if any problem were to arise, that would appear in subsequent generations, whereas if you only did this with males, then any negative effects of performing this technology would only last for one generation. So that's been a proposed idea or limit by some bioethicists. However, it's been generally acknowledged that this technique is, as far as we can tell, safe and reliable, and there are no negative consequences of doing it as far as we know. But those are the sorts of things that were considered prior to this being approved. Okay, I'm going to go through the second recent reproductive genetics or reproductive biology advance in recent years, and that's the cell-free fetal DNA testing. Now, in week 1 or 2 we talked about prenatal genetic testing by chorionic villus biopsy or amniocentesis, and I mentioned at the time that there's something called a non-invasive prenatal testing, and this non-invasive prenatal testing is a way of detecting fetal DNA in maternal blood. Now, within maternal blood during pregnancy, there is cell-free DNA floating around, and that just comes from cells as they die. A lot of your immune cells undergo programmed cell death and they just die, and you have DNA floating around in your blood. And we call that cell-free DNA. In a pregnant woman beyond a certain date, about 20% of that cell-free DNA that you find floating around in the blood is actually of fetal origin. How it gets there, we're not really going into the details on that, just be assured it does get there. In the same way that proteins from the fetus get into maternal blood, and we can test for things like alpha-fetoprotein and so on. So it gets there. What we can do is look for it, and that will give us the genotype of the fetus, along with a background of normal maternal DNA. So by using some very clever quantitative DNA sequencing and quantitative polymerase chain reaction approaches, we can quantify the amount of fetal DNA, and we can find out whether that fetal DNA comes from the mother, comes from the father, because what we can do is look at the mother's genotype and then look for paternal DNA and use that as an idea to see what is the likely genotype of that fetus. Okay, so the simplest scenario is where the fetus we want to test whether the fetus has received a mutation that we know is present in the father but is not present in the mother. So this could be something like Huntington's disease. If the mother is negative for Huntington's disease, the father is positive for Huntington's disease, if you can detect that Huntington's disease mutation in the mother's blood during pregnancy, you know that the fetus is going to have the genotype for Huntington's disease, and that therefore we can give that information to the parents and they can terminate if they wish. So that's a relatively straightforward and simple way of doing that. So it really helps if the father is a different genotype to the mother. What's more complicated is if you want to try to detect something like chromosome duplications in cell-free fetal DNA. So CFF DNA is cell-free fetal DNA, whereas CF DNA is cell-free DNA, as in total. So this is more complicated because we've got maternal's cell-free DNA and cell-free fetal DNA as well. And some of that cell-free fetal DNA is exactly the same genetic sequence as the mother's DNA, and then you've got thrown in there into the mix some of the father's DNA as well. Now in a couple of slides I'm going to go into some complicated genetics about how this works, but suffice to say it works. And what is now being done is replacing some of the karyotype and fish-based invasive methods for taking a amniocentesis and coronic villus biopsy. So in the past, the only way we could do this is take a coronic villus biopsy or amnio and perform FISH for chromosome 21, look for chromosome 21, 18, 13 and sex chromosomes to look for all common chromosomal abnormalities, or we could do a full karyotype, and this is quite laborious and invasive. Now what is being offered is women with a high chance of Down syndrome are offered this non-invasive testing, and this is on the basis of maternal age, paternal age, or raised blood markers such as alpha-fetoprotein, and maybe also if you've seen a defect on an early scan as well. It's known that this detects greater than 97% of all Downs fetuses and can also detect Edwards and Patau syndrome and sex chromosome abnormalities. And by introducing this non-invasive test, we can reduce the number of coronic villus biopsies and amnios by thousands per year. You've got to remember that there's a chance of spontaneous miscarriage by doing a coronic villus biopsy or amniocentesis. We completely get around that problem doing this non-invasive testing. Okay, this slide is really quite complicated, and if you don't follow it, probably don't worry too much. But it explains the theoretical levels of DNA doing one of these non-invasive prenatal tests, the blood tests, looking at chromosome 21 for Down syndrome. So let's take the scenario we've got maternal blood, non-pregnant. The cell-free DNA will be equally split between one chromosome 21 homologue and another chromosome 21 homologue. And we can look for regions on chromosome 21 that differ between this woman's maternal and paternal chromosome 21. We can tell the difference between them because there are subtle differences between everyone's two copies of chromosome 21. So just by focusing in on the regions that we know differ, we can say half of the cell-free DNA in that woman's blood, half of it relates to one of the chromosome 21s, half of it relates to the other chromosome 21s for any given location. Now if that woman is pregnant, we know that 80% of the cell-free DNA will be her own DNA, equally split between homologue A and homologue B, and 20% of it will be from the fetus, equally split between one of her chromosome 21s and the father's chromosome 21. So when we quantify all of the chromosome 21 material in this pregnant female, we would expect to see the following. Now firstly, 10% of that chromosome 21 DNA will be paternal. It will match the father's genotype. The other 90% will be made up of either 40% or 50% of A and 40% or 50% of B. And if this is 50%, this one would be 40%. If this one's 40%, this one would be 50%. So that would tell us that, you know, we could have the situation where we have 40% maternal A, 50% maternal B, 10% paternal. That is what we would expect to see for a normal pregnant woman. Now, if we have a situation where we're looking for Down syndrome, and let's assume that it's the maternal chromosome 21 that has been duplicated, when we do the maths, this is what we actually see. We would see maternal A 53%, maternal B 40%. Now we know maternal B is going to be 40% because 80% of the DNA is going to be maternal, half of it should be, let's say, maternal B. What we've got here is the additional 20% that is the fetal DNA. Now that should be made up by one of the chromosome homologs and the paternal. And if we've got an imbalance here, that they're not equally split, then that would suggest that there's too much maternal A chromosome 21 versus the paternal chromosome 21. So there's, in this case, an excess of maternal A chromosome 21. So that's suggestive of a chromosome 21 duplication, and it's the maternal A. If it was the father's chromosome 21 that was duplicated, this would jump up to 13.3% instead of 10%. So 10% is a normal situation, and it would just jump up to 13.3% instead. And that would be diagnostic to say that we have got a Down syndrome case here where it's the male chromosome 21 that is duplicated. So this can just be shown sort of as an image here of what fragments of DNA would look like if they were, you know, coloured differently in maternal blood. If we've got maternal blood with a normal fetus, I call it euploid, we've got all of these blue fragments would be from the mother, and all of these orange fragments would be from the fetus. Now in this example it's talking about 10% of fragments. It's a maximum of 20%. It can vary from 20% downwards. So in this visual example, the fetal DNA, cell-free fetal DNA, is 10% of all of the DNA in the blood, and that's shown as those orange bits. If you've got a fetus with chromosome 21 trisomy, you get a few extra bits of DNA that are chromosome 21, and they will be overrepresented. Now in reality it doesn't matter which chromosome is duplicated in Down syndrome, whether it's maternal or paternal, but maybe this one illustrates how it can be done much more simply, where this shows, imagine if you're going to take all of the cell-free DNA from some mother's blood, sequence all of those bits of DNA and assign them to all of the different chromosomes. Now based upon the size of the chromosome, this graph is going to be higher, and if we assume 20% of the cell-free DNA is coming from the fetus, you would get, you know, you can say, okay, all of this is maternal DNA and these bits here are fetal DNA, and you can sort of tot them up. But even without knowing which way round, you know, whether this is fetal or not, the total amount of chromosome 1 DNA would be this many different bits, and chromosome 2 is a bit smaller, so you get fewer bits associated with chromosome 2, and so on until we get to chromosome 21 and chromosome 22. Chromosome 21 is one of the smallest chromosomes, and you know, let's say this is what you would expect from a normal pregnant female. If this peak compared to chromosome 20 is proportionally higher, so let's say it just goes from here to a little bit above to a certain threshold, then we can say that the fetus is likely to have an excess of chromosome 21 material. So, you know, this is purely sort of pictorial illustration of what would be going on, but we'll be looking for the total number of chromosome 21 sequence fragments to be higher. So the ratio between, let's say, chromosome 21, 20 and 21 will change, with chromosome 21 material being more abundant relative to chromosome 20 than a normal individual. So if this is a little bit too high, that suggests that chromosome 21 is duplicated somehow in that fetus. Okay, this is all using very complicated technology. It's using quantitative sequencing, which you have not yet covered, or quantitative PCR that you have not yet covered. So don't worry too much about the technology. You just have to accept we are finding more chromosome 21 material in the maternal blood than we expect. Okay, so that summarises everything that we've covered on this part of the module, this bit of reproductive biology and genetic testing. We've covered everything from human imaging, where we've looked at ultrasounds, we've looked at biochemical tests in week 1. In week 2 we've looked at prenatal genetic testing, for where we are looking for things like Down syndrome or inherited diseases by taking a chorion or villus biopsy. Week 3 we looked at pre-implantation genetic diagnosis, where we can select for disease-free embryos. In week 4 we've looked at how we can create embryos which are free from mitochondrial diseases by doing those 2 methods that we've just looked at today. And then finally we've looked at non-invasive prenatal testing by taking maternal blood and effectively doing the test on maternal blood that would have been done on the chorion and villus biopsies. It's not the same test, it's far more complicated, but it's giving you the same result. Okay, so that's completion of this subpart of the module.